Hey everyone, it's me again. Today we're gonna finish the packet finally. All right, we're gonna continue with DNA. All right, this is the last day. Let's finish the packet. Let's get it out of our lives. All right, so please listen. Make sure your volumes are turned up and listen close. Here we go. So, today we're going to talk about the nitrogenous base pairs. You may be wondering, what is that? Okay, so the DNA double helix, right? That ladder that's twisted that contains all of the genetic information that we got from mom and dad, right? So there's adenine, the one with the A right there, here, and then there's thymine, guanine, and then cytosine. Okay, and they pair up specifically, right, with its type. So thymine always goes with adenine, and then guanine always pairs with cytosine. All right, and they never pair with anything else. Okay, except when there's RNA. So when there's an RNA strand. Okay, so RNA is formed in the nucleus, right? It exits the nucleus into the cytoplasm, and then it goes to the ribosome, all right, where it helps to make proteins, all right? So the thing with RNA, ribonucleic acid, is that instead of thymine, you see down here, it's got uracil, okay? So when it's DNA, it's got thymine. All right, so it's adenine, thymine, cytosine, and guanine. The RNA has everything except the thymine. That thymine is now uracil. Okay, so just remember that. Now, let me go back. I'm going to get out of here. We're going to come back to this slides. All right, don't worry. But I'm going to go to the packet. So here we are. Okay, so we already talked about DNA. The 4 nitrogenous bases, right? They're right there, adenine, guanine, thymine, and cytosine, like we talked about. And they pair up specifically, right? One with the other. And so thymine never goes with guanine, and guanine never goes with adenine. Okay, so I pretty much did number 1 for you, did number 2 for you, and then number 3, which base does cytosine pair up with? I already basically gave you that one also. Okay, so there you have it. Now, question number 4. All right, this is where it gets interesting. So, based off of the DNA double helix and how they pair up, they're asking you to finish the strand. Okay, this is the template. All right, so this is the one that's being printed off of, right? So remember, it's always the opposite. So what's this one going to be? Okay, thymine. What am I going to put here? That's right. And this thing's not even working. Okay, let me see if this thing is gonna work. Let's see. Why is it not working? There we go. All right, just kidding. All right, so it's all sloppy now. Okay, so I'm gonna put the A there, right, for the adenine. Excuse the sloppiness. All right, and then there's adenine, so then I'm gonna put the T, right, for the thymine. All right, and then what goes with the guanine? Cytosine, so I'm going to put a C. All right, and so you're going to keep going with that. So adenine, okay, and then the thymine. And you're gonna do the rest, all right? And it's super easy, all right? It's almost a no-brainer, all right? It's simple. So let's say we're going to do an R and A strand. Hopefully it comes out. Let me see. Okay, there it is. It's all sloppy, sorry. Okay, so let's do an RNA strand. So as you already know, let's go back. Thymine is now replaced with uracil. All right, so you already know adenine and thymine go together, right? So the A and the U are going to go together now instead of the A and the T. All right. So if it's an A, so now you know what? Uracil is going to go there. So let me put that uracil. There it is. Okay. And then there's thiamine here. What am I going to put here? That's right, adenine. Okay. And so same thing. Okay, G. So the C is going to go there. Uh-oh, we got an A. So don't put thiamine. What are we going to put? Uracil. And what these are, so every 3 of these codes for a particular protein. All right. So remember, the DNA contains the commands, right, that we need in order to have our physical features. So each 3 codes for one particular thing, and I'll show you that in a little bit. So let's finish this one. So we got thiamine. What's going to go with thiamine? Okay, adenine. And then cytosine. What's going to go over here? G, guanine. And I got a T, so adenine. All right, let's stop there. All right, so that's the RNA strand. So all we're changing is the thiamine for the uracil. All right, so I'm gonna let y'all complete this strand right here. And if you want for fun, you can put an RNA strand there and finish that one as well. All right, but just remember that every 3 of these codes for something in particular. And we'll see that in a bit, right? Like that. Okay. So those 3 go together, those 3 and these 3. All right. So I'm pretty much doing this for you. So please follow along and make sure you put them in, because you do have to turn this in, right? All right. So number 5. So what's all this stuff here? It looks all crazy. Don't worry. I'm gonna explain it to you, and you'll get it. All right. You're all smart. So, which DNA strand is used to make RNA? So it's asking for a strand. Okay, so underline that right there. So we've got DNA here, right? So this is the same DNA double helix that you see up here. Same thing. It's just that now it's unzipped itself. And it's starting to make it's not making RNA. RNA is popping up in here, right? It's coming from the nucleus, and it's going to join the template strand, and it's going to make it's going to transcribe, right? Remember transcription? So remember that RNA comes from DNA, and then proteins come from RNA, right? So this polypeptide here is just a protein, just a fancy word for a protein. And that comes from what? The RNA, right? So you might be wondering, well, how in the world does this RNA just kind of decide to hop in here? Well, let me show you. It's something called an RNA polymerase. Let me make that bigger. All right. So this RNA polymerase, and you're right, remember, it ends with an ASE. So that means it's an enzyme, right? Polymerase. So this enzyme goes in there, and it helps that RNA to start making that code. All right. I'm not going to go too into depth with this, because then I don't want to confuse you and add too much information. But just so you know, this is extra. This isn't in the packet. But the RNA polymerase helps that RNA to join that template strand. All right. So it unzips the DNA that we see here, right? It unzips it, this RNA polymerase, so that the RNA strand can join here and start making its code, right, from the template strand. All right, sounds a little complicated. It kind of is, but you'll learn that later. Don't worry. Okay, so let's go back to the packet. So which DNA strand is used to make the RNA? So looking for a strand. So we've got the RNA right here. So which one is helping to make that RNA? There it is. I'm not going to say anything other than that, but you're welcome. All right. I basically gave you the answer for number 5. And then number 6, what will the transcribed RNA, so remember, RNA is made through transcription. Okay, so what will the transcribed RNA be used to make? All right, so then what's the next step? What is this transcribed RNA going to make? This polypeptide, which is a what? It's a protein, right? So I'm pretty sure you already know what you're going to put there, right? All right, so let's move on. So remember that that strand we were making, that RNA. So thymine, adenine, and uracils. Remember that RNA strand does not have thymine, right? What does it have? It has uracil instead, right? So let's go back and see. So there it is right there. Okay, we're making an RNA strand from the coding strand, right? Okay, so let's go back. All right, so let's check to make sure. Here we go, we got it right. Yeah, we're doing good. All right, so just to make sure, right? All right, so there it is. So remember, every 3 is making a particular code. Okay, so that's why I'm underlining each 3 to kind of just separate them. All right, but the RNA strand, remember, uracil, right? Let's go to question number 7. Oh wow, this chart looks kind of complicated. It's not. It's so simple. All right, I'm gonna go through it with you. Don't worry. Okay, so number 7. Does this chart work with DNA or RNA? Well, let's look at it. We already know that DNA has adenine, guanine, thymine, and cytosine. This one has got that U. What does that mean? Okay, I'm going to leave you with that, all right, for number 7. I'm not going to say any more because I don't want to spoon feed you answers, okay? So it's obvious what it is, right? So it's asking you, is it DNA or RNA? I already pretty much gave you the answer, right, without really telling you, but I hinted at it. All right, so question number 8. The first 3 letters in the RNA strand is AUG. So let's look. So there it is, AUG. That one code. It's called codon. All right. They're not telling you that here, but I am. All right. It's called a codon. These 3. Now the chart tells us this gives us the amino acid methionine, right, which is a start codon. So this code means, hey, start doing your job. All right. So let's check it. All right. So A. First letter, let's see, A, and then U, and then, so A, U, and where's the G? Okay, so A, U, so this is the second letter, and the third letter, G, so it's got to line up over here, okay? So it says methionine. Where is methionine? I don't even see it. Oops. It's not even here. Oh, there it is. Sorry. Just kidding. Okay, so so you do the first letter A, U, all right, and then in the same block right here. In this same block, we're going to look for the G. There it is right here, and where they all meet is right here. I'm going to just put a red circle. Boom. All right. So A in the first, U in the second, and then G. There it is, methionine. All right. So that's how we're going to figure out which code it is. So scientists, biologists spent so much of their time figuring this stuff out and made this chart for all of us to point. There it is. There's the answer. So they did us a great favor. All right. So what does the second set of 3 letters AUC code for? So let's look up here. So now we're coding for this, the AUC. All right. Let me just go right there. Okay. We got A. Okay. U, which is right here, and then C. Where's C? Right. Okay. There it is right there. So isoleucine right here. All right, let me make a red circle so you can see it. There it is. All right, so A in the first letter, U in the second letter, and then in the third letter, it's a C. Okay, right here. So boom, we got it. All right. So I did number 8 for you. You're welcome. Now let's do number 9. Okay, what does the third set of 3 letters code for? Let's look over here. UCG. Okay, so we're doing those. I'm going to write them here. So U, C, G. Come on. All right, there it is. So UCG. So now we got to find what polypeptide that one is. So you tell me, what is it? Well, let's see. First letter U, and then you got a C, and then G. Right there. There it is. Serine. Okay. So the first letter, you're always looking in this one block. Second letter, right, and then third, right, and you're staying in that one block from the first letter. Okay, so that G is right there. So serine. All right, so we did number 9. Now number 10. What does the last set of 3 letters code for? So we got 3 more to go. UAA. UAA. All right, let's see. Let's find it. All right, U. Let's go to the first right here. All right, first letter. And then let's go to the C. And then it's, oh, sorry, am I doing it right? All right, sorry, A. I was looking at number 9. So U, A, and then A. It's a stop. It's a stop codon. So here we are stopping the code. Hopefully that came out right. Did I spell that right? Yeah, stop. So it's a stop codon. All right. So number 7 was basically a, number 8 is a start. Sorry. So number 8, methionine is a start codon. So it starts it, and you know what? Stop. So with that being said, I am going to stop also because we are done with the entire packet. Make sure you put in all the answers like I did right here, and you turn it in, folks, and that way you won't have any homework to do this weekend. Please don't turn in any late work. All right, turn it all in. And that's pretty much it, folks. Have a great weekend. Be safe. Hey, that the coronavirus is still out there. It's decreasing. It is, but be safe. Social distance yourself. Wear your mask. Don't gather in large groups. Wash your hands. All right. I'll see y'all Monday. Y'all be good. All right. Peace out.